Hello, and welcome back to my genetics lab. Today we will be using these two DNA strands to walk through transcription, translation, and discuss point mutations. We want to end up with 4 amino acids, so first we have to find a start codon and a stop codon that will leave us with 4 amino acids. The start codon we are looking for is ATG, and the stop codons can be TAG, TAA, or TGA. Let's look at our DNA sequences closely. The only place where we will get 4 amino acids is if we start at this ATG start codon here, reading from the 5' to 3' direction. We then have to scan to find our stop codon, which is right here, TAA. This top strand is our template strand, and this bottom strand is our coding strand. Now with these codon sequences, we can start transcription and write out our mRNA strand. Now that we have written our mRNA strand in the 5' to 3' direction, we can start translation. We have to use this chart to find our amino acid sequences, which end up being methionine, valine, serine, lysine, and the stop codon. Now we are going to review some point mutations that can take place in our cells. These 4 different types of point mutations are missense, nonsense, read-through, and silent. Let's first define missense, then see an example. Missense is when the point mutation changes the amino acid. So let's take this AGC and instead say there was a point mutation to give us AAC. This would completely code for a different amino acid, which can alter protein function. Instead of serine here, we get asparagine. This mutation changed this G into an A, which would be a transition because we changed from a purine to a purine. Next, let's define nonsense. A nonsense mutation is when the point mutation changes the codon so that it becomes a stop codon. For example, look at this AAG codon. If we change this to UAG, then we get a stop codon and end up with only 3 amino acids. Here we changed this A to a U, which is a transversion because we changed from a purine to a pyrimidine. A read-through is when the stop codon is changed to a codon that codes for an amino acid, resulting in a longer protein. For example, let's take our stop codon and change it from UAA to UAC. This instead gives us tyrosine, and we would continue coding for other amino acids in this direction until we hit another stop codon. We changed this A to a C, which is a transversion since we changed from a purine to a pyrimidine. Finally, we have a silent point mutation, which occurs when the change codes for the same amino acid. Let's take our GUC and change it to GUU. They both code for valine, so there is no difference. Since we changed from a pyrimidine to a pyrimidine, this is a transition. Thanks for stopping by my genetics lab today.